Hello, everybody. Good afternoon, good evening, and good morning to attendees throughout the world. This is the third of our WSPID webinars, and still focusing on COVID. And now the most important recent development are the COVID-19 vaccines coming to light. And we have an amazing webinar in store with two excellent speakers, Professor Miguel O'Ryan and Professor Walter Orenstein. We will be starting off with Miguel O'Ryan, who's also the chair of the WSPID International Scientific Committee, and you can see his titles and qualifications on the screen. We are very happy that you've all joined us. We already have a list of questions, challenging questions, and please use the Q&A module for additional questions, and we will do our best to answer. The duration of the webinar will be round about 90 minutes, with round about an hour for our two speakers, and then question time. So without further ado, I'm going to ask Miguel O'Ryan to give an overview of COVID-19 and vaccination. Thank you very much.
Thank you very much, Mark. It is truly my pleasure and an honor to be in this session, especially sharing with whom I would say is today one of the world champions in vaccines, that's Walter Orenstein. And what I will try to do in the next 20 to 25 minutes is to set the stage on vaccine development. To come next maybe to the more important part is to see how we're going to use the vaccines, what we're going to do, and what are the tremendous intense problems and issues that we will have to deal with and challenges that we'll have to deal with in 2021. So I've been participating actively in several committees in Chile, advising for COVID 2019. So my potential conflict of interest, I'm one of the co-PIs for the Janssen vaccine trial in Chile that is stopping enrollment today. So I assume that most of you are in the medical field or in the biology field, so this is not strange for you, but I think it helps to understand where we want to go with vaccines. Basically, remembering that coronavirus is a common virus structure with its protein capsid. Within it lies the RNA, and the spike protein that protrudes is critical because it is this spike protein that attaches to the ACE2 receptor that allows the entry of the cell, together with interactions with other proteins. And clearly, there's no doubt that this has been the main target for vaccine development. Once inside the cell, the mRNA is liberated into cytoplasma, is replicated, and at the same time is translated by our ribosomes to produce more viruses. And these viruses make it out of the cells to be presented to the antigen-presenting cells, the main driver, probably the orchestra director of our immune system, where it presents the different antigens that is presented to himself, to the other cells of the immune system, mainly the B cells, T cells, and the helper cells, producing neutralizing antibodies or cell cytotoxicity, among many other immune mechanisms. Very important is having memory cells, which of course are key not only for protective immunity in the future after infection, but importantly after vaccination. So this is the key player, the antigen presenting cell, and it helps us then to advance to understand the different platforms that we can see here. And I like this Nature paper that came out in April because I think it's quite illustrative to understand the different platforms. starting from the most traditional platforms that we all know, which are the inactivated virus vaccines and the attenuated virus vaccines, which basically inactivated through chemical or heat processes, usually it's chemical, or attenuated through changes or modifications in the genome. So the whole intact virus structure is presented to the antigen presenting cell. This is a very traditional vaccine strategy. If we would have to make a simile, we could think of oral polio and inactivated polio. Advancing a little bit more, we have the virus-like particles, the VLPs, which also present several proteins to the antigen presenting cell. This is using, as you probably all know, synthetic or synthesis of these proteins through expression systems and auto-assembly of the virus particle in these VLPs. This strategy is used for papillomavirus today. And this is one of the platforms then that is advancing. A little further on, always in the concept of introducing proteins is platforms derived on viral proteins, mainly the spike protein, but also there's some strategies related to the M protein. And this strategy has also been used in the past, the simile, and we're using it. It can be hepatitis B vaccine. And then we move to what can be considered the relatively new platforms because they have not been used out of clinical trials or early use in some less massive diseases such as Ebola, which is the concept now of not introducing directly the proteins that we are interested in, but is introducing the genomic sector, the genomic part of the virus that codes for the protein of interest. And this can be introduced in several ways. One is using viral vector as a Trojan horse, where we introduce to the genome of that vector, which usually are adenoviruses, but it could also be measles virus or VSV virus or other. And this can be, this segment of the spike protein then is introduced, integrated into the genome of this adenovirus. So when this adenovirus is introduced into our cells then produce the spike protein. These viral vectors can be replicating and non-replicating, just as the real virus can be inactivated or attenuated, following that logic. The great majority of the developments today are non-replicating adenoviruses. What are the potential issues that we have with these platforms that need to be addressed? And you all have heard about vaccine disease enhancement, a concept where vaccinating individuals and then being exposed to the real pathogen actually enhances disease due to an incomplete immune response, not completely neutralizing and deriving an immune response actually to Th2 responses or antibodies to actually enhance the entry of the virus to cells. This was an issue with RSV in the 60s, and it has been conceptually seen feasible for SARS viruses, and therefore all the vaccines have to have passed the reasonable evaluations to intend to discard the possibility of vaccine disease enhancement in preclinical trials. And to make a long story short, the signals are looking pretty good in this sense. Another factor is anti-vector immunity, especially relating, well, to the vector-related vaccines, where using a virus actually can induce an immune response to the Trojan horse, to the vector, so that the second dose actually could have an effect by producing immunity to that second booster dose, reducing the impact of the booster dose. So this is something that also has to be dealt with, and so far it seems that the candidates are being able to overcome this possibility. There are many interesting issues and developments that have occurred in terms of this, especially when maybe the half-dose effect that we saw with with the Oxford vaccine. And the need for adjuvants is also very common with, especially the viral proteins, the VLPs, and adjuvants are important to strengthen the immune response, but at the same time, they tend to increase the possibility of vaccine-related side effects. So that is an issue also needs to be looked at. And then we have, following the strategy of using genetic material of the coronavirus, either RNA or DNA, now not in the form of a vector, a viral vector, but using lipid particles for the RNA vaccines as a way to enter this RNA into our cytoplasm, where the viral proteins are finally produced to be exposed or developed in the antigen-presenting cell to expose then the proteins in the surface. Those are the RNA vaccines. And then we have the DNA vaccines, which are a little bit more complex, because in order to introduce the DNA not into cytoplasm, it has to go into the nucleus of the cell. So either through plasmids or through using this specific device of electroporation, this strategy has advanced to try to introduce then this genetic material. So these are the main strategies, and the potential issues related with RNA vaccines and DNA vaccines, I would say, are basically the fear of the new, because these vaccines have not been used, as I say, programmatically worldwide, but actually they're not so new. These have been developed since the 1990s, and several thousands of individuals have received these vaccines. And then the potential issues of genomic integration or anti-DNA antibodies specifically for DNA vaccines, or enhancement of autoimmune diseases have all been discussed related with these vaccines. But we could say today also that these vaccines are looking very, very safe. Important to say that most of the genetic material after introduction is destroyed or eliminated. And the integration, which is commonly discussed, has not been seen to be an issue with RNA vaccines for clear, and not even for DNA vaccines. But of course, these are issues that still will need to be looked at. So now that we know which are the basic vaccine platforms, let's see what the overall development is until the day before yesterday. Starting with the inactivated vaccines, and this is the WHO page, which is fantastic because it really is renewing itself every 1 or 2 weeks, and it's amazing how the candidates increase day by day. And we can see here the ones that are in phase 3 trials, and in light blue, here we see the inactivated vaccines in phase 3 trials, mainly coming from China and from India. Then we see the vector-based vaccines from Oxford. Here we see also from CanSino from China. The vaccine from the Gamaleya Research Institute and the Janssen vaccine. So those are all in advanced phase 3 trials, as you all know. And then we have protein subunit vaccine from Novavax, and we have a more recent one from the Chinese Academy of Sciences. The 2 RNA vaccines that have received a lot of press, Moderna and BioNTech, Pfizer. And newly into this group of phase 3 is the Medicago vaccine, which is one of the few VLP vaccines, because there are not as many VLP vaccines as there are from the other candidates. And of course, we have a few in phase 2 that will most likely be advancing to phase 3 quite quickly. And here there are some that are doing very interesting studies in different areas, CureVac, Arcturus, and others. And then we have the long list of vaccines that are in phase 1. One would have said before last year that, okay, we don't even need to look at these because it'll take 3 to 5 years to advance or to think about clinical development. But now we're talking actually 2 or 3 months. Years have come down to months. So many of these that are now in phase 1 are probably rapidly advancing to phase 2. And we probably will be seeing them advancing to phase 3 within the next months or so, within 2021. And there are 161 other labs around the world that are developing vaccines in preclinical stages. So the efforts have been absolutely astonishing. So it's interesting to show what is the data that we have so far from these vaccines. And please bear with me that this may not be absolutely complete, because basically it's based on PubMed reviews and other reviews to try to see what data is there for the vaccines. But we have data, not much yet for Sinovac. There's a trials data that's how the trials are going to be done, but we're expecting to see results from Sinovac hopefully in the near future. The same from Sinopharm, where we have early studies. And then we have the data from Oxford AstraZeneca, where we have the reports from phase 1-2, and I'll show you what just came out. We have studies interesting from CanSino. You can see all of these are published in the main world journals, Lancet, JAMA, New England. We have data from Gamaleya. There was a lot of discussion initially about showing the importance of this vaccine without providing data. Well, data came out in Lancet in September, very small numbers, of course. We'll have to see if hopefully more higher figures are provided. Janssen so far has provided its phase 1-2 trial, but in medRxiv, still to see where it's going to be finally published. We have Novavax published in New England, and Moderna and Pfizer with their preliminary reports, Moderna and Pfizer with these 3 papers. So there is amazingly fast and quick publications coming out, but it's all still quite preliminary. And the latest data we have, at least in information that one can get, are the press releases. We have the BioNTech Pfizer press release with that literally said that with around 53,538 subjects, there was about 90% efficacy for symptomatic disease from day 7 post-dose 2. For the mRNA 1273 from Moderna, the press release was that there was more than 30,000 with 94.5% efficacy for symptomatic disease, with no severe disease in vaccinees, 11 occurring in the placebo group. And then we have the press release from AstraZeneca reporting that with around 11,600 enrollees within Brazil and UK, of which 2,700 received half of the first dose because of an issue occurring there, there was an overall 70% efficacy from day 14 post-dose 2, with this interesting potential difference of 62% for the full dose and 90% for those receiving the half dose in the first dose. And finally, we can say that as we speak, information is coming out, and here I must thank my friends in the audience who especially, well, he there who knows about information just coming out. And this one is the information that's being presented to the FDA on the Pfizer-BioNTech vaccine. Now we have 60 pages in this document, and there's plenty of information to take decisions. This also was presented to the authorities in the UK. And I just show one slide here, or one table of the many tables that shows the vaccine efficacy of around 95%. And look at the distribution of cases, 8 and 162 in all participants. Importantly, also showing high efficacy in different age groups. And this has also been looked at in individuals with different conditions and other situations. So there's a lot of information now obtained from this vaccine that's being analyzed tomorrow by the FDA. And in terms of side effects and adverse events, there's also very intense presentation of the results. I just do a quick summary here. Intent is not to go in depth, but these are reactogenic vaccines. The most common solicited adverse events were injection site reactions, 84%, fatigue, 63%, headache, 55%, muscle pain, chills. and about 14% having fever. The good thing is that severe adverse events were okay and were comparable between vaccinees and placebo. So looking good. We'll have to see then how we're going to roll out with these and other vaccines in the future, as Walt will discuss. And as you all know, this came out yesterday, is the Lancet paper on safety and efficacy of the UK Oxford vaccine. And also here, just to present one piece of data, one of the tables. This was mostly efficacy and severe adverse events. You can see overall here a vaccine efficacy that seems to be around 70%. There are also these interesting differences that were shown. 90% in the one that used the low dose, standard dose. But look at the confidence intervals there, it is overlapping. So this will have to be reviewed intensely to try to see if this, which seems to be important, how important it can be for the future. And in terms of side effects, across all 4 studies, the vaccine had a good safety profile, and serious adverse events also were not concentrated in one or other. Maybe some small issues there that will be worth discussing, but overall, this is also looking very good. So with that, my final comments would be that the world has high hopes, I would say as never before in vaccines, and the vaccine community in general, from developers to researchers around the world, has responded, I would say also as never before. The efforts here have been absolutely amazing. And certainly we will see more developers presenting submissions soon. And so we'll have a lot more data there to look at, to compare and to analyze. And here things will, of course, will get interesting, will get complex, and we'll have to make decisions. There will be differences in data. We have to look at those carefully. The outcome variables used by the different studies, we'll have to see how they compare or not compare, not to jump quickly to erroneous conclusions. We'll have to see, is there any differences in reactogenicity and efficacy profiles between the vaccines, analyze them the best as possible to see how we can provide the best recommendations. So future studies, including placebo-controlled trials, will need to be discussed and considered and most likely continued, as was discussed just a couple of days ago by the WHO ad hoc ESCR group as published in the New England Journal of Medicine. So this will also be a huge challenge for the world and for the vaccine research community. And finally, vaccine trials in children also will need to be discussed. And I'm sure this is an issue that Walt will bring into discussion in his talk too. So how we move from trials to the real world, what will be the rollout? Certainly it's a huge challenge, and we'll start to listen on how this will be thought upon, at least in the US and in the world, with the talk that I'm looking forward to from Walter Orenstein. Thank you very much for your attention.
Thank you very much, Miguel. I'm now going to introduce my co-moderator, Professor Eben Adeyujigbe from Nigeria, chairperson of the Nigerian Infectious Diseases Pediatric Society. Over to you.
Thank you very much, Mark. Thank you so much, Miguel, for the presentation. So we are having the next speaker, the second speaker, Walter Orenstein, and he is a global icon in vaccines. He is Assistant Director presently at the Emory Vaccine Center in the United States, the former Director of the United States Immunization Program, and he's a former Assistant Surgeon General of the United States. He has worked, with the Center for Disease Control and Prevention for 26 years on immunization. He has also been the chair of the World Health Organization poliomyelitis technical consultative team. And he has vast experience. Actually, I think he is the man of the hour because even his research interest is vaccine hesitancy and how to improve uptake, which is what we are in, considering what level the world is with the COVID vaccine. Thank you very much. I hand over to Walter. Please take over the presentation. Thank you, sir.
Thank you very much. Thank you, Miguel, for giving such an outstanding presentation to lead off. My focus today deals with how do we optimize the public health impact of COVID-19 vaccines as they are rolled out. Since I am based in the United States, I will try to use our efforts as an example of what might be done regarding rollout vaccines to achieve optimal impact. And I am a member of the Scientific Advisory Board for Moderna, which is making one of the messenger RNA candidate vaccines. My favorite line is vaccines don't save lives, vaccinations save lives. A vaccine dose that remains in the vial is 0% effective regardless of what the clinical trial showed. And our goal is to get the vaccines into the bodies of people for whom they are recommended. So what are the topics I want to cover? One is what things will we not know through the trials and what will be needed to be determined in the post-licensure or post-availability period. Number two is insights into how vaccines will be allocated and used, particularly the issue of emergency use versus full approval, the risk groups for whom vaccines might be indicated, and when they might get to the general population. Another issue is the need for assessing vaccine effectiveness on an ongoing basis in observational studies, and the importance of continued vaccine safety monitoring. Another one is how realistic is it to achieve herd immunity? What will it take? When can we stop our non-pharmaceutical interventions, such as masking and social distancing? And finally, to talk a little bit about the COVAX initiative being led by Gavi, the Vaccine Alliance, and trying to get vaccines to low and middle income countries. Be unanswered. One is priority groups for vaccines, because we don't necessarily have all groups in there, and how do we determine those groups? A second, and I saw that in many of the questions asked, is the duration of immunity. We're not going to know the duration of immunity when we're talking about months of data leading to availability of vaccine. The third issue is risk factors for vaccine failure, which might indicate more doses, and the concerns about waning immunity that I just mentioned above. The fourth issue is potential rare causally related vaccine adverse events and risk factors for those events that can be made potential contraindications for vaccine. We're getting lots of data on protection of individuals from disease, but we are not getting data for the most part on the impact of vaccine in reducing transmission, which is important. We're all assuming it will reduce transmission, but we don't know that for sure. And finally, barriers to vaccine access and acceptance. How do we overcome vaccine hesitancy, and how do we remove other barriers to access? The National Academy of Medicine in the United States was asked to derive or develop an allocation framework. And this is what they came up with. The goal was to reduce severe morbidity and mortality and negative societal impact due to the transmission of SARS-CoV-2. So what should be used in deciding allocation criteria? One is who's at risk of acquiring infection. Another factor, who develops severe morbidity and mortality. A third is maintaining societal function and what would lead to a negative societal impact. And a fourth issue is focusing on who's transmitting to whom and trying to reduce that transmission. And they came up with 4 allocation phases. And they looked at things like ethical principles, such as getting maximum benefit, equal concern, the mitigation of health inequities, and fairness, transparency, and trying to use evidence as best as possible in deciding the allocation framework. This is a slide I got from the CDC, the Centers for Disease Control and Prevention in the US website, shown here that I accessed in early December, looking at the rates of hospitalization and death by age group. And they used 18 to 29-year-olds as the comparison group. And what you can see here is very clearly the older the age, the higher the risk, particularly of deaths. And for children, lower. So that's why most allocation schemes do not have children as high priorities on the allocation scheme. And as I said, they came up with 4 phases of allocation. The number one group at phase 1A, the jumpstart phase, was high-risk health workers and first responders. Healthcare workers are number one on virtually all the schemes I've seen for prioritization. They mentioned number 2 as phase 1B of persons at all ages with comorbid additions, and then phase 1, also older adults living in congregate or overcrowded settings. In the United States, our advisory committee on immunization practices has recommended that phase 1 will include the high-risk health workers and older adults living in long-term care facilities. Phase 2 was key staff, such as teachers and school staff, child health workers, critical workers in high-risk settings, and I'll show you that in a minute in terms of who they might be, people with comorbid and underlying conditions that place them at moderately higher risk, homeless shelters, some with mental illness and a variety of other things, and people in prisons and jails, and all other adults not included in phase 1. In phase 3, young adults, here they get to children, and workers in industries and occupations important to functioning of society, and then eventually everybody in the United States. They also made a number of recommendations for programmatic use. One is to leverage and expand the use of existing systems, structures, and partnerships across all levels of government to provide the necessary resources. So that, for example, we have efforts to vaccinate adults in the United States. Let's piggyback onto that infrastructure with COVID-19 vaccines. For countries that don't have much of an adult program, to reach out in other ways to use existing infrastructure rather than having to create a whole new infrastructure to get to adults. Another factor is, as I mentioned, to remove all barriers to access, and one of them is out-of-pocket costs. The government should take over covering all of the costs so that there are no out-of-pocket costs. Number 3 is the need to fund research, not only on developing vaccines, but research on how to get the benefits and risks adequately communicated to get people for whom they're recommended to accept. Also resources, once you have that knowledge, to launch a vaccine promotion campaign. And again, as we learn to adapt that and modify that campaign to obtain ideal acceptance. Another issue for the US was the recognition by the National Academy of Medicine that we are one world, and that if we simply focus on our own population, we, for humanitarian reasons, aren't helping others in the world. And it's even for our own domestic health security, because this virus got to us by importation. And we need to have supplied for the rest of the world. And they mentioned dedicating a portion, for example, 10% of supply we would normally get to global allocation. And the COVAX facility would be the key group to do that and to engage with the World Health Organization in doing that. And for those interested, you can obtain this full report at this website. It was released in, I believe, around August, but is available for free, as I understand it, on this website. Now, one of the big issues is it looks like virtually all of the vaccines will not be given full approval. They will be given emergency use listing or WHO approved allocated vaccines. In the US, it's emergency use authorization, or EUA. And what does that mean? With each EUA decision, our Food and Drug Administration, our regulator, weighs the potential benefits of a product against known and potential risks. And we've used that before. The big issue here is we're having now about 2,000 people dying a day in the United States from COVID-19. And if we wait for the final process, which can take months and months, we may get more and more dying. And so the issue here is to potentially put out a vaccine before full approval. And what would be the efforts to do it? One, the results are very positive, but perhaps the manufacturing process has not been fully looked at. The results are positive, but the FDA has not finished reviewing hundreds or thousands of pages of documents. Is the UA status low quality or substandard? No, when supported by sufficient evidence. And how would clinicians handle it differently? The healthcare advisors must be informed about the benefits and risks, and they must discuss them with patients. Requirements, I think, would be problematic within EUA vaccines, but again, it's important to give information that can allow potential recipients to make an informed decision, including what their risks are if they decline administration. The need to overcome hesitancy is a problem. In the US, we use the Operation Warp Speed term to show how we were moving so fast. And it sounded great at the time, but it backfired. It gave the feeling to many in the public that corners were being cut and that the vaccine was not safe. And we need to assure that we are getting safe vaccines. There's also conspiracy theories circulating about collusion of government with drug companies and lack of an understanding of what an emergency use authorization or listing really means. So what do we need to do to overcome vaccine hesitancy? We need to understand the reasons, and as I said earlier, to fund research to determine those reasons. We need to fund research into how to overcome hesitancy. When I was director of the US Immunization program, my chief of communications said you need the right message, delivered by the right messenger through the right communications channels. What we have seen in childhood vaccination with regard to overcoming hesitancy, the right messenger is often the primary care provider. And so we need to equip those primary care providers with the information they need to overcome hesitancy. And again, much of the hesitancy, as I just said, is due to a perception that the vaccines are not adequately being tested. And so we need to have information to show how much we've done to test them. Well, one of the things I said is we will need to have continuing efforts to evaluate vaccine efficacy or effectiveness in observational studies. We have several types that are being considered, and the World Health Organization, and I am on a committee of that, is considering putting out a document recommending various ways to assess vaccine effectiveness in observational studies. There are at least 5 different kinds of methods that are being considered. One are cohort studies, which could be prospective, where you look at a population where you've been able to roll out vaccine and a population where you haven't and look at their incidence of disease and safety issues. Or they can be retrospective when you look at, after a period, people who got vaccine versus people who didn't get vaccine and what their incidence was. Then there are traditional case control studies where you have cases and then you match them by age, by gender, by other things, and you look at the vaccination status of your cases and of your controls, and also you could look at safety issues in the 2 groups. A test negative design has become very popular for influenza in particular, as well as other vaccines. And this, I think, may be the most strongly recommended. And what is done here is to take people who test positive as your cases and look at their vaccination status, and then test negative for your controls and their vaccination status, and calculate odds ratios and vaccine effectiveness. The screening method was developed in the 1980s as a rapid way to try and determine whether we needed to do a more detailed evaluation of effectiveness. What's done is to look at the vaccine status of the cases you have and compare that with measures you have of the overall vaccination status. And in the UK in particular, they've advanced that to a much more sophisticated level, but it's a way to do that. Another one, which I'm not familiar with, but which has been mentioned, is something called regression discontinuity design, where, for example, a vaccine is recommended for persons 65 and older. You may use as your control group patients 55 to 64, so they're close in characteristics, and then look at their experience and their incidence rate. And one of the things that's absolutely critical to these studies is to get good documentation of vaccination status. And what that means is not relying on verbal history, if at all possible, and having good documentation of which vaccines people got and when. So just to go over in a little bit more detail, the cohort studies can be prospective or retrospective. Prospective, there are some ethical considerations if in fact a group is considered for vaccine, but if, for example, if rollout is done geographically, you might have your control cohort in an area not initially allocated compared to an area where it is. Its results can easily be communicated to policymakers. You can not only estimate effectiveness, but you could also, in your control cohort, estimate what the actual burden of COVID-19 is. The weaknesses are the need to be able to illuminate these cohorts and assure that they're comparable in characteristics and that there's no confounding. For retrospective studies, you need to enumerate vaccination status independent of COVID-19. You need to assure cohorts have equal access to diagnostic tests. And again, there may be substantial confounding in healthcare seeking behaviors, and there is expense. It's more expensive, particularly if you have large cohorts. For traditional case control studies, it's more efficient than cohort studies, but you have smaller numbers and less cost. You can focus efforts on finding cases instead of following large populations, and you need fewer non-cases than you do in cohort studies. The problems are misclassification of vaccination status, although that's probably greater than compared to cohort studies, selection bias, who are healthcare workers going to test? Are they more likely, for example, to test unvaccinated, which would tend to inflate vaccine effectiveness, and choosing controls comparable to characteristics of cases to minimize bias. The test negative design, it does minimize bias in healthcare seeking behavior of vaccinees because all cases and controls seek care for comparable illnesses. They seek care at the same facilities, decreasing differences in access to vaccines in different communities. The vaccination status is usually obtained before laboratory tests are available. And a key question is if test sensitivity and specificity is high, you should minimize misclassification. The weaknesses is healthcare workers might be less likely to test vaccinees. Will vaccination status ascertainment be accurate? Will the test be adequately sensitive and specific? Should specific tests be recommended? If tests are done early in illness, might test positive patients be more likely to seek care later as their illness intensifies than non-cases? And will vaccinees more likely to have underlying illnesses which are exacerbated with respiratory illness, leading to an increase in vaccination of non-COVID cases and a potential misestimation of effectiveness? The screening methods I mentioned, it's markedly reduced expenses since vaccination status of the community is available from other measures such as surveys. You don't have to collect data since the surveys presumably are existing. The weaknesses of the survey data may not be represented of the population for which cases are collected, and vaccination status data may come from administrative data rather than from surveys, which are less likely to be accurate. So to go over what Miguel covered in his talk, what do we know about safety? We have, for the most part, 3 phases of clinical trials before vaccines are released to the public. Phase 1 is 20 to 100 volunteers, and that looks at gross safety, immunogenicity, if there are serious side effects, and looking at dosing and what's the appropriate dose. Phase 2 is several hundred volunteers, looking at the most common side effects and how are their immune systems functioning. And phase 3s are hundreds or thousands. What we saw in the prior presentation by Dr. O'Ryan is that you can get, in our studies of COVID, 15 or 20,000 vaccinees with roughly equal numbers of non-vaccinees. And these are the trials needed for availability, and they give you general safety and they give you effectiveness, but they don't necessarily estimate effectiveness in subpopulations that might have lower efficacy, and they don't necessarily determine safety in these same kinds of subpopulations, nor do they determine long-term safety issues. So what do we need to assess safety in the post-licensure setting? One is before a vaccine is rolled out, it will be helpful to collect background rates of various clinical syndromes to potentially be compared with rates following vaccination, so that you can immediately begin assessment of causality versus coincidental. It's important to work with vaccine providers and healthcare providers to report adverse events following vaccines. Often this may not be helpful in causation as much as setting in place hypotheses to be tested in more rigorously structured scientific studies. It's important to have systems in place to assess rates of clinical syndromes post-vaccination to compare with background rates. And finally, to have committees of experts to evaluate whether events following vaccination are likely to be causally or only coincidentally related to vaccines, as done with many data safety monitoring boards in the clinical trials efforts. And this is just some examples in the US that the Food and Drug Administration, our regulator, and the Centers for Disease Control and Prevention, the CDC, use. One is the Vaccine Adverse Event Reporting System, or VAERS, and this allows anybody to report. It could be a healthcare provider, it could be a vaccinee, it could be a parent of a vaccinee. And again, it analyzes the data. It can compare with the reports of other vaccines to suggest there might be problems. It could look at clusters of specific events, but generally is not that great for evaluating causation. And that's where something called the Vaccine Data Link, or the Post-Licensure Rapid Immunization Safety Monitoring Network, uses PRISM. And what this allows you to do in many people is to calculate rates of a given specific clinical syndrome in vaccinees and non-vaccinees to compare them. And it also allows you, if there is causality, to look at specific groups for which the risk is there, whereas other groups it's not, and those groups may get contraindications. And then we have a system working with universities to do special studies if we feel they're indicated. The work group that for the vaccine allocation system in the US, the Advisory Committee on Immunization Practices, considered the following criteria in making their allocation decisions: ensure safety and effectiveness of COVID-19 vaccines, reduce transmission, morbidity, mortality, minimize disruption to society and economy, including maintaining healthcare capacity, and ensure equity in vaccine allocation and distribution. And there were things that they looked at that they realized they didn't know. So they don't know everything about the epidemiology and immunology. For example, how important are children in transmission, even though they seem to be at lower risk of severe disease? Nevertheless, they have to make their decisions based on what we know, but be prepared to change them as more information is known. We know certain things about the data of safety and efficacy, but we don't have ideal data and efficacy, and we need to take that into account. And we don't know the number of doses. Almost all of the trials have 2 doses of vaccine in them, or at least 2 doses of vaccine. There are a few that have single doses. And that means we need a system to assure people get those doses and data to show how effective a single dose is. It turns out in the paper that Dr. O'Ryan showed from the AstraZeneca Oxford trial determined an effectiveness of roughly 50% for a single dose, and almost about 95% for 2 doses. But at least that is helpful, that if you fail to get that second dose, you at least might derive some protection. Remaining information gaps in certain populations, again, risk of disease and severe outcomes, vaccine safety and efficacy, the transmission dynamics, and initial data would be helpful in informing prioritization. But as in clinical decision-making, it's rare you have everything you want at the time you have to make your decisions. There are 3 things that the committee considered: science, implementation issues, and ethics. The science was the COVID disease burden, what we know about vaccines, and balancing benefits of harms. Implementation deals with values of the target group. How feasible would it be to implement? I mean, you could make a great recommendation, but if you can't get vaccine out to them, then that's a problem. And ethical is to maximize benefits and minimize harms, to promote justice, minimize health inequities, and promote transparency. And in the US, the goal, as I said, is healthcare personnel, about 21 million people. And then the next group is residents of long-term care facilities, which is about 3 million people. So that there's still a lot of people that are lower priority on the initial rollout. This just gives you some examples of what we're thinking about with healthcare personnel, hospitals, long-term care facilities, outpatient home, pharmacies, people in public health, essential workers such as food and agriculture, adults with high-risk medical conditions, shown here, chronic obstructive pulmonary disease here, sickle cell disease, and then other adults who are elderly who might be living in a variety of places. So in summary of their work group, number one were healthcare workers. As of November 21st, they had confirmed 228,000 COVID-19 cases in healthcare personnel, 822 deaths. COVID-19 exposure could occur inside and outside, resulting in absenteeism, which is a problem in the infrastructure, quarantine, infection, and illness. And so vaccination has a potential to reduce absenteeism, and long-term care facilities demonstrates more cases and deaths averted by modeling in vaccinating staff than vaccinating residents if you have to decide between the two. Implementation could be easier. We already have a system to get influenza vaccine, and you can go to hospitals, for example, to get them. And many of them have the equipment needed to keep vaccines at appropriate storage conditions, such as with the mRNA by Pfizer BioNTech that has to be stored for long term at -70 degrees centigrade. And the ethics of preserving healthcare services not only benefits the healthcare workers, but really the entire population. Now, the last thing I'll talk about is the COVAX facility. This is led by Gavi. It has the Coalition of Epidemic Preparedness Innovations, the World Health Organization, UNICEF, the Pan American Health Organization, and Multilateral Development Banks. The CEPI is supporting research and manufacturing scale up. Gavi is a group that is coordinating this, pooling procurement, incentivizing manufacturer expansion, securing supply, and giving advanced market commitments. The World Health Organization is providing normative guidance, vaccine policies, safety, regulation. UNICEF is coordinating procurement and distribution across the various COVAX participants. PAHO is doing that as well in the Americas. And then multilateral banks are financing support directly to participants. And this is the goal of COVAX, to end the acute phase of the pandemic by the end of 2021, to deliver 2 billion doses to low and middle income countries by the end of 2021, to support the largest actively managed portfolio of vaccines globally, to offer a compelling return on investment by delivering COVID vaccines as quickly as possible, and to guarantee fair and equitable access for all participants. And this just gives you an example of thinking. This, again, is not necessarily what will happen, but to just give you an example. So they would give advanced market commitments to AstraZeneca for 300 million, Sanofi, GSK for 200, Serum Institute of India for 200, and other vaccine candidates. And by the end of 2021, guarantee these manufacturers they had a market and end up with 4 billion doses available if possible. And these are just some of the things that the Advanced Market Committee is doing, is to accelerate the availability of vaccines. Here they're talking about 1 billion doses by the end of 2021, thus making vaccines affordable, guaranteeing these large markets, and protecting healthcare workers, the most vulnerable population, decreasing transmission, hopefully around the world, and restarting the world's economy. And then finally, herd immunity. Will we achieve it? We all want to achieve it. Vaccines protect in two ways: one, inducing active immunity in recipients, but indirectly in reducing the potential exposure of susceptible individuals who can't be vaccinated or fail to make a good response when they're surrounded by immune individuals. And this just shows you some herd immunity thresholds from a variety of diseases with basic reproduction numbers, which is the number of people who would be infected if you lived in a 100% susceptible community on average. And the CDC in the US estimates that the best estimate for COVID herd immunity is about 65%, with a range of generally 60 to 70%. The basic R0 level is the average case in a fully 100% susceptible population would give rise to 2.5 cases. So in summary, critical factors to be considered in COVID-19 included allocation include those most vulnerable to severe disease and critical infrastructures, especially the healthcare infrastructure. It is important to take advantage of existing infrastructure for vaccine delivery, including cold chain and means of reaching populations not often covered in immunization programs, particularly adults. Older adults seem to be at greatest risk of severe COVID. Likely priority groups include healthcare workers and adults in long-term care facilities. It is unclear at this stage what priorities will be given to vaccinating children. Studies of COVID-19 vaccines are in early phases in children. It's critical to have ongoing strong systems to assess vaccine effectiveness and safety, and the COVAX facility is critical to getting vaccines for low and middle income countries. Thank you.
Thank you, thank you, thank you. Thank you so much. I see a lot of questions and comments coming in. So thank you so much, Walter. Thank you for the presentation. You just answered some of the questions. So some of them were on children. Yes, they don't have a severe infection most of the time, but they are carriers. And people are worried that would they be vaccinated at this time, or would they have to wait for their own clinical trial before they can be vaccinated? In the same vein, how about the pregnant women? Questions have come. What happens to the pregnant women? Are they going to be vaccinated now? Or as usual, they have to wait? So those are the questions that I want to bring to the fore for now.
I think those are important, and Miguel may have better information. I think, to me, we need trials in children. We need to determine what the appropriate dose is, what the dosing is, and we need to have safety and data on effectiveness. What would be great, but I haven't seen from the current clinical trials in adults, is if we could develop a correlate of protection, such as an antibody level, which would greatly facilitate doing trials in children where you wouldn't have to wait to see who becomes ill and who does not become ill. But I think we need that. I've been pushing. I'm glad they're starting, but I think they need to be moved more quickly into there. I think the big issue in children, even if they're transmitters, is that with the more sick people and the more people who are suffering, the initial allocation will not be based on a herd immunity threshold, but on a protection threshold. We won't have enough doses to do a herd immunity threshold to begin with. And so the focus on vaccine will be on people who are suffering most or people who need to maintain our critical infrastructures. But I do think eventually children need to be covered. Miguel, you may have.
Yeah, I would just like to add, at least in my mind, and I think most of us, as infectious disease people have in mind respiratory viruses and the role of children in transmission, which is very important. But I think that, the epidemiology is showing that for this particular respiratory virus, the role of children is less. And the transmission is mainly young adults who are getting infected and transmitting to others. Now, does that mean that children are not important? We do not know fully, I think, mainly because most of the children of the world were taken out of circulation very early on, with a tremendous impact of limitation of viral infections in general. So, but it seems that they do not play such a significant role. So I don't think their children, in being a pediatrician, I say this, are not a first target for vaccination. If they will be a target at all, I think is something that we will need to try to solve, getting better info. And for pregnant women, definitely, I think there's some vaccines that probably will not be able to be used in pregnant women. We would have to think about probably not using mRNA vaccines or DNA, I don't know. Maybe Walt can think about this. Maybe inactivated viruses could be used in pregnant women, but basically, in general, I would say probably pregnant women would not be a main target for vaccination, at least in the initial phase, and we get more data, and for some of the vaccines.
I think that I would agree with that. It's a very important question, and there certainly is risk in pregnant women, but I think as Dr. O'Ryan said, it's a lot easier to deal with vaccines we know a lot about than the initial candidates out of the gate are all vaccines we've had very limited experience with.
Another question is with the mRNA virus vaccines and the potentials of causing problems, later in life. What is your advice? Because some of these vaccines are going to be mRNA, and people are wondering what will happen later. Immune problems, anaphylactic problems. So, what is your problem? What is your solution or your advice for now?
There was actually, that was brought up in the New England Journal of Medicine, and you could look at I forget exactly when, it's a series of letters to the editor, and a person did bring up the concern as how long would messenger RNA last, and the authors used an influenza mRNA study that said it would not last long. I'm not aware of any data with regard to SARS-CoV-2 in mRNA, but the presumption is that it wouldn't last long. But we will have to follow people, and we will have to do those kinds of studies. I think what's very impressive for the two mRNA vaccines that we've heard about, the Moderna and the Pfizer-BioNTech, is those efficacies are fantastic. I mean, in the US, the FDA was willing to consider a vaccine with a point estimate of efficacy of 50%. Here we've got in the 90s. And in essence, when we get enough doses, if in fact it does reduce transmission, and let's hope so, most of our vaccines do reduce transmission, not only protect the individual from disease, we may quickly be able to achieve herd immunity thresholds, or at least really tamper down transmission very quickly with these vaccines.
I would just add, I'm not an expert, and I know there may be basic science experts that know exactly how the integration and interactions may be, but looking at the data that's out there, I think that mRNA vaccines, it's important to not have the myth that mRNAs can be integrated easily into our genomes and produce abnormalities that lead to cancer or diseases, related with genomic alterations. Because mRNAs, basically RNAs, stay in the cytoplasm and are rapidly destroyed. And that has been shown, there's data. So another thing can be DNA vaccines. And there was a little bit of concern about the possibility of integration of DNAs using plasmids into our DNAs, but also because these technologies have more than 10 years. We actually, there's been out there since the 1990s. So there is a lot of studies out there. And so far, these concerns that were raised initially are not standing. I think that they're looking reasonably safe, and we should reduce our worry about a potential alteration of the human beings' genomics materials because of the use of these vaccines.
Yeah, I think Mark tried to answer one of the questions online. And I also had Walter refer to it about the microchips in these vaccines. So can you shed more light on that?
Well, that's part of the problem with the conspiracy theories. Clearly, there's no microchips being injected into the vaccines. And this is where I think it's exceedingly important to look at, to get the people who are trusted by the people who aren't trusting, be they religious authorities, be they primary care providers. What's needed is to develop coalitions where the true data can be presented and to get that out to people. And I think we need to be prepared for that sooner rather than later. I mean, these issues should be addressed with a Q&A and perhaps even research focus groups beforehand. My director of communications, I gave one quote. I used to say, with regard to the media, if it bleeds, it leads. And we know when we're vaccinating millions of people that there are going to be some very bad things that happen immediately after vaccination. And we need to be able to explain to people they may be coincidental and not causal. Paul Offit, who's a very famous vaccinologist, tells a story of his wife, who was a pediatrician, who was about to vaccinate a baby, and the baby had a seizure just a few minutes before the vaccination. If that had occurred for a few minutes after the vaccination, clearly would have been blamed. And so we need to try and get ahead of these kinds of things that are going to happen.
I'll ask one more question and I'll hand over to Mark. Just to find out, people are worried. A lot of money has gone into production of these vaccines. And I think Mark said many governments are also trying to ensure that the vaccine is available for their countries. But we also know that many low and middle income countries would not be able to afford it. So what's the plan? Rolling out the vaccines to where it is most needed, at least for once.
Well, that's where the COVAX facility is most important. That's what Gavi, the Vaccine Alliance, is trying to do. And I got these slides from Ann Lindstrom, who's a senior official in the immunization program at the World Health Organization. So they do have substantial funds. And what they are trying to do is these advanced market commitments so that companies assure that the lower income and middle income countries can have access to the vaccines. Now, one of the big issues is getting the vaccines to the national level doesn't get it out to the peripheral level. And there needs to be investments also in getting the vaccine out under appropriate cold chain mechanisms and the like. But the goal is to make this affordable and free to people. And so the manufacturers wouldn't be making any extra money than they would be in their initial production and what the COVAX facility would pay them.
Thanks. I'm going to ask a few questions. The one is strain variability across the world and the vaccines. Do you have any, either Walter or Miguel, any thoughts about this question?
Yeah, you know, I think it enters into the arena where we don't have all the answers, and we will have to see what happens in the future. But at least in my mind, I think with the experience that we have so far, we know that this virus, like any other virus, mutates, and we have, you know, hundreds, if not thousands, of different virus mutation types at the moment circulating around the world, with a prevalence of maybe a few in some places that make clades or specific types. But at least I have not seen, it's not been reported, there's been hints that maybe some change is related to more transmission or to more disease severity, but that has not been sustained epidemiologically. Maybe a little bit in the lab, but not epidemiologically. So far we have a year of this virus, millions of people affected, and we're not seeing that this virus is changing in a significant way. Now, will having heard a lot of people vaccinated help selection of maybe a virus that can escape vaccination, you know, due to a change in the spike protein? It's possible, but I don't know what you think, Walt, but I'm not seeing this at the moment to be a big risk. Certainly, this is not influenza. It's not a virus that changes as influenza that needs to be changed every year. It has a different biology. So we'll have to wait and see, but I think overall, maybe a one-time, maybe a booster dose sometime later, but not repetitive boosters will be needed for this vaccine. But we'll have to see.
I would agree completely. I think there are two issues that lead to continued recirculation. One is your own body loses its immunity over time, and you become susceptible because you've lost that immunity. The second, as was mentioned, is mutation in the virus to evade the immune response. One of the good things about this is most of the vaccines are targeted at receptor binding site and hopefully a more conserved area of the virus. We don't know that. We will have to see. But at least at this point, we've not seen, as Dr. O'Ryan said, escape from antibody. We've seen some people who've had natural disease develop a second infection, but those are anecdotes and appear to be exceedingly rare. And we need to be careful about over-interpreting anecdotes. Basically, we've seen people, the vast majority of people who've been infected seem to be protected, at least for some period from that. And so we're just going to have to look and adjust if we have to. One of the advantages of some of the vaccine technologies is if there is mutation, they potentially could fairly easily be adjusted to give us a new vaccine if we need it. But hopefully we won't. And I think I agree completely with Miguel. The data I've seen do not at this point support that as a concern.
Thank you. Could you comment on the strategy of screening for antibodies prior to immunization? Could that be a way of rationalizing limited supply? Is that a strategy that might be considered?
I don't know, Miguel, you may want, I'll take my public health background. The problem, I think, with testing for antibodies is the more complicated you make it, the more difficult it is. My experience in public health as opposed to clinical medicine is a one-size-fits-all approach is far easier to implement giving multiple different things. If this happens, do this, and that happens, do this, et cetera. In fact, that's one of the reasons why in the US we went to a universal influenza vaccination program, because we were having so many groups that people had to figure out who to give it and who not. I think the issue here is that if you have to get a blood test, you'll either have to have a blood test that's available at the clinic, or you'd have to send that blood test out, get it run, get it to come back. I think it gets more complicated. And so certainly in the US, they're saying no, and even they're recommending vaccinating people who had COVID in the past, since we don't know for sure. And so they recommend against doing serologic testing prior to vaccination.
Yeah, I agree. I mean, if we didn't have enough vaccines, then we would have to start thinking about this and see how we would do it. But at the moment, I think it wouldn't be practical.
Thank you. We've had a number of questions about allergy, some about egg allergy, and others about the Pfizer warning for people with an allergic tendency or diathesis should avoid vaccination. Is there any, could you comment about that? Or both those concerns?
It's a good question. I haven't seen, I mean, it's almost like, you know, if you're very allergic, difficult to eggs, well, with egg-related vaccines, of course, but then why should that transduce to some of these vaccines that are not related to eggs? But then, yeah, there are a lot of particles and proteins in these vaccines that maybe people that are extremely allergic to different antigens could have a severe reaction. We don't know. But it's a good question. I don't have a straightforward answer. We would have to see what the data shows to see what the recommendations is. I think that the companies tend to go on the safe side here, and they want to try to avoid, based on maybe on the lack of information, if a person that's very allergic could have a severe allergic reaction to one of these antigens. But that's all I can say. I don't know what you would think.
Yeah, and I think that what I don't know, maybe you know, Miguel, is whether people with such reactions were excluded from the trials. But if not, certainly we've not seen very bad allergic reactions in the trials that I'm aware of to date. And these are not small trials. I mean, the Pfizer trial is over 40,000 individuals, so over 20,000 vaccine recipients, and the Moderna is over 30,000, so over 15,000 vaccine recipients, at least with those two vaccines.
Thanks. I'm going to hand over to Eben for any more, if you have any more questions from the panel, from the audience.
Okay, then thank you. There are a few more, quite a lot of questions. So, but one of them is that for example, in the UK now, the vaccine is being given out to people, and it's contraindicated for women who are in the childbearing age and lactating women. And yet, many of the health workers are women in that age group. So the person wanted Walter to please shed some light on this. It's contraindicated, and yet most of the people who are health workers who are at high risk are in this age group. So how do you reconcile the?
Well, if in my mind, that the healthcare workers are important, of course, because they've been infected in more frequency, not necessarily in higher severity. They are higher risk due to the age. The high risk is the elder, no, and as age goes up. But now the issue of vaccinating women in pregnancy age, I think, is an issue that for a programmatic use, and Walter maybe can answer this better. It's like for any other vaccine that's not recommended for pregnant women, is that basically different to the clinical trials where you do a testing of the woman with a pregnancy test to assure that she's not pregnant. You cannot do that in the real world setting. So you have to rely more on the person who finally decides to get vaccinated and to warn her that the data on the effect of the vaccine in pregnancy is unknown. So probably you don't want to take that risk. So you want to take the preventions. But I think that's what you would have to do. And then if you do start having, and we will have, pregnant women that are vaccinated, we start getting information on what happens with those pregnancies, of course, over time, which will provide additional safety data. And that will be important. But Walter, the strategy there.
I couldn't agree more with you. I think the issue to me is, I remember when rubella vaccines were first rolled out, since these were live attenuated viruses, there was big concern. And what happened is women inadvertently were vaccinated. Let's say became pregnant shortly after, or didn't know they were pregnant at the time they were vaccinated. And we collected that data and saw no risks. So we made it easier. I think what's very important here is the monitoring systems that need to be in place, including where women inadvertently who turned out to become pregnant or had been pregnant at the time of vaccination but didn't know it are collected and look at what happens with themselves and with their babies.
So, one more question, because of in some places, because of influenza outbreaks and everything, they are having to give everybody influenza vaccines. So, what is your take? Should they at the same time also give people the COVID vaccine while they are given the influenza vaccine?
And this goes to the issue of vaccine co-administration, which is a very interesting issue that has not been studied. So that is data that we don't have there. So I would say that at the moment, you do not want to co-administer vaccines because we don't know if there can be, vaccine interactions. But that's something that will also have to be rolled out. That would be my take.
I think to me that would be the issue, but it would be very important to have a system in place, such as a reminder and recall system, to bring the person back for the vaccine they missed at this time. I think it's going to be very important in every country that the NITAG, the National Immunization Technical Advisory Group, really look at these issues and come up with recommendations for their country. And I would bet that the World Health Organization, the Strategic Advisory Group of Experts on Immunization, or SAGE, will probably address that as they develop sort of model recommendations.
So hand over to you, Mark. Thank you.
Okay, thank you. Could I ask our speakers to speculate on Africa, where the epidemiology often seems a little bit different to what's seen in the US and in Europe and in Latin America, and what your thoughts about vaccines for Africa are?
I'll let Walt start.
I think they should get vaccines. The issue is I just don't know the epidemiology that much, but what I do feel is it makes a lot of sense as vaccines become available to get the healthcare worker community vaccinated. I think it's extremely important in maintaining the infrastructure because, as I said earlier, it benefits not only the healthcare worker, but it benefits everybody else whom the healthcare workers might take care of. So I think that would be critical. I think the other one as to which of the other high-risk groups are most critical. Is it residents in long-term care facilities, people with comorbidities, et cetera? I think that to me, it would be important to collect data, if at all possible, because Africa is not homogeneous. It may vary between places within Africa, is which groups seem to be most at risk, and then try to focus vaccines towards those groups.
I would say probably Eben would be the best person to help us answer this question, coming from a country where there are huge cities. You know, of course, we're all interested in trying to understand. We're all extremely worried that this virus could really cause a huge toll in Africa. And it doesn't seem to be the case. And there's always speculation, is it because it's not being registered adequately? But it doesn't seem to be that. Is it related to the different age distribution? Because actually the age span is lower, so the risk groups for severe disease that we see in the more Western world is not there. Probably there's something like that. But with this environment in Africa, I completely agree with Walter that vaccination will be also very important and critical, but the decision making of the age groups then, or the priority groups, will probably have to be focused on the epidemiology that our African colleagues are finding. And I would worry, especially in the big cities, where you have millions and millions of people. That's where COVID-19 really hits very hard.
Thank you. I'm going to conclude with a very important question from Susan Mayring, who points to the high reactogenicity of the COVID vaccines and compares it to influenza that's usually quite mild. And what is one's approach to this? Surely there's a huge education campaign that needs to roll out if this is the case.
I fully agree. I fully agree that the education campaign here is very important. The reactogenicity of vaccines does not seem to have all the same profile. You have to be careful with the comparisons, but the ones that seem to be at the moment more reactogenic than others are the mRNA vaccines, then maybe followed by the viral vector vaccines, and the least reactogenic will be the inactivated virus vaccines. Maybe, we'll see. And if we're going to have several vaccines in one country that have different reactogenicity profiles and maybe different efficacy, hopefully all will be acceptable, of course, to be approved. But that has to be conveyed. That information has to be conveyed in a difficult way, but in a transparent way. And that, to me, is critical to assure that people will get vaccinated. Because if you start vaccinating and 10% of fever, or 15% of people start having fever or sick leaves, as Susan says, and we don't anticipate that can bring huge problems in the rollout.
I would agree. And I think the issue here is, again, trying to develop communications based on what it would be to people to explain, because I think for me, it would be putting in context with what would happen if I got COVID-19. And maybe that will work, maybe it won't. I think it would be very important prior to rollout to begin with the knowledge we have and dealing with people who might be hesitant and working out with them what seems to be the information that would convince them to do it versus would not allay their fears. So I think it would be, again, a very important thing to deal with different groups. And again, as I said, Africa is not homogeneous, nor are the countries in Africa homogeneous. And we need to do it in various subpopulations within Africa to try and get understandings of what people think about this and how to weigh the risks and benefits.
I would just add, it wouldn't just be an issue for Africa. This is an issue for all over the place, all over the world. We need to convey the correct messages. This will be the huge challenge for what's coming in 2021 for the rollout.
I agree. Thank you very much. So thank you very much to our two great speakers, Walter Miguel, my co-moderator, Eben, and our coordinator and education manager from Kennis, Hester. And this will be available on the website. And please, if questions are unanswered, contact Hester and she will distribute. We'll try to get the answers. And everyone stay safe and look after yourselves and your families. Goodbye.
Thank you. Bye-bye.
Thank you.
Thanks.